All right, class, good morning. This is Mr. Clancy. We are going to focus on today's, it is Tuesday, March 24th. And I am going to focus with you on the something called RNA, which is ribonucleic acid. And you'll see this is used in protein synthesis. So basically, I want to go through these notes on chapter 13. You'll find a copy of this in the assignment section. The copy of the notes will be in the topics section. And then there's an assignment for you, which is basically like a graphic organizer between DNA and RNA, so we can get used to how these two are contrasted. How are DNA alike and RNA alike? How are they different? And how are they involved in this magnificent process of protein synthesis? That's going to be our focus for the rest of the week. So let's go through some of the notes with it. Basically, I want to start with just going to go through these. You can take these kind of stay with me as we go through the notes, and then when we're done, you can complete the assignment. RNA basically stands for ribonucleic acid. Ribonucleic acid. All right, and the sequence of bases in DNA. In other words, on the strand of DNA, how the sequence basically stands for what we call a code. I'm going to make this a little bit skinnier on my pen. I want to make this somewhat thinner for you. All right, let's see how that goes. We'll stay with the blue, huh? What do you think? All right, and we'll see that, in other words, the sequence of bases. DNA is comprised of nucleotides, which you've learned about, which are basically which mainly are constructed of the phosphate group, the sugar, and the nitrogenous base. Every nucleotide has the sugar and the nitrogenous base, whether it's DNA or RNA, or excuse me, everyone has the sugar and the phosphate group. The only difference is the nitrogenous bases, and it's the order of these nucleotides that actually makes DNA different. And also the order is very, very important in specifying the instructions for making proteins later on, which we'll learn more about tomorrow. So basically, RNA is involved in the cell's coding instructions. Coding instructions. All right. And then RNA is basically comprised of building blocks, which are called nucleotides, very similar to DNA. All right, each consisting of a sugar. This sugar, by the way, is called ribose. We'll go to that in a couple of minutes. A phosphate group. Or just a phosphate, if you like it, a nitrogenous base. All right, and then three key differences between DNA and RNA, and this will have a lot to do with your assignment, so you want to make sure you're paying good close attention here as we do this and taking some real good notes for reference. RNA is single-stranded, whereas DNA is double-stranded. Remember, DNA is like the spiral staircase. All right, RNA contains the sugar, which is called ribose, R-I-B-O-S-E. So you can think of ribonucleic acid, ribose, whereas DNA, deoxyribonucleic acid, contains the sugar deoxyribose. All right, and then RNA contains a base, a little different base, which is called uracil, or just sometimes dubbed U, all right, the first letter. And this is in place of thymine. That's a key difference between DNA and RNA. All right, and some other differences that we'll see. DNA always remains in the nucleus. Well, RNA goes from the nucleus to the sites of protein synthesis. You might remember this from first semester. These are called the ribosomes. And remember, these can be kind of floating around in the cytoplasm, or they can be kind of embedded on the side walls of the endoplasmic reticulum. And remember, that's called the rough ER, rough endoplasmic reticulum. That's what makes it rough, the embedded ribosomes on the side of it. So basically, the proteins are the site of the protein factories of the ribosomes. And RNA's primary function is protein synthesis. Protein synthesis. And proteins are very important from a structural standpoint and a functional standpoint for us both. Synthesis. All right, it does this by carrying out the instructions held by DNA. So DNA can almost involve, if you can imagine DNA as sort of like the boss, right? The plant manager sitting in the ivory tower, right? Telling everybody else what to do. And the RNA is sort of like the molecular slave, if you will, for DNA. Good way to remember it. All right, now there's 3 types of RNA. I'm going to explain what these do briefly. And then we're going to see this in a protein synthesis activity, which I'm going to have you do at your house later on in the week. So the first one is called messenger RNA. Messenger RNA. Now sometimes that is dubbed little m and then uppercase RNA, mRNA. All right? These molecules basically carry information from the nucleus to other parts that should be part. My Boston accent kind of slipped in there a little bit, of the cell. The second one is called ribosomal RNA, or R, little r, then big R, big N, big A, okay, RNA. These basically form the most important functional, in effect, they make up a ribosome, which remember is the protein factory. All right, and the third one is called transfer RNA. Transfer RNA, or as you guessed it, it is dubbed tRNA, little t, big R, big N, big A. And in effect, this carries coded, if you will, instructions to the ribosome. And matches them to the ribosome, matches them to the mRNA message. All right. So this process, mRNA, in RNA synthesis, all RNA is basically transcribed. From DNA. All right. And so the process where RNA is made is called transcription. It's a message. It's orders. It's a record. So transcription occurs in the nucleus. All right. Segments in effect, in transcription, segments of DNA will serve as templates. All right, to produce complementary RNA molecules. Next page of our notes. And then during transcription, there's an enzyme called RNA polymerase, kind of like DNA polymerase. All right, binds to DNA and in effect unzips, or kind of splits that ladder, right? Kind of cuts this ladder right in two, okay? So I, in effect, have two the DNA strand, the DNA double strand, is split. The unzipping process is splitting the bond kind of right down the middle of the molecule from splitting that DNA from one double-stranded molecule into two single-stranded molecules. This is similar to DNA polymerase. During DNA replication. All right, and then the RNA polymerase binds to the DNA at kind of areas, sites, or specific regions, and these regions are called promoters. We'll see more about this in a video later. Promoters, in effect, show the RNA polymerase where to exactly start making the RNA. So RNA basically helps with doing that. And there's also a process called editing. Some RNA molecules require some editing prior to use. In other words, just like you'd edit a tape, if you would, or edit I shouldn't say a tape, a video. All right. Portions of the RNA are cut out, and these portions are called introns. INTRONS. All right, and the remaining pieces of the strand are put back together, and these are called exons. And the exons basically end up being the pieces of DNA for that. So you can view that site on that. This isn't the only site. You can copy and paste this. You'll see some examples of introns and exons as we do that. So the main lesson today I want you to see is DNA and RNA. Now, a couple things. There is a short video that I want you to watch this video. I want you to fill in the notes, go through the notes with that, and then also complete the assignment. Please do your best on the assignment to fulfill it on your own first. There is an answer key on the end to help you with that. Don't just copy the stuff from the answer key. You're really not helping yourself if you do that. So that is today's lesson. I'm praying for each one of you. I hope you're praying for me. And also a special prayer, if I could ask, for my wife and my daughter. My wife and daughter are nurses, and they are on the front line in the battle against coronavirus. My wife works at the VA hospital, and they are dealing with veterans. They help the veterans who have served our country, giving them the care. If you could pray for her today, I would really appreciate that. I pray for protection for her, that she would not contract the virus and ultimately give it to me. Hopefully that wouldn't happen. And the same with my daughter also, that she would not. My daughter is a nurse at Children's Hospital, Lurie Children's down in downtown Chicago. I pray she could help with that as well. One other prayer request, if I could. There is an organization that we serve, as you know, at Wheaton Academy, Kids Alive. One of the sites at Kids Alive in the Dominican Republic in Santo Domingo is really having a hard time right now. The school is closed down. The school is really kind of their hope for the future for these young children. But also they're having a really difficult time getting enough food for everyone to eat. So if you could remember that in prayer too, that would be great. All right. Love you all. I look forward to seeing you as soon as possible. So do your best in the assignment, submit it back, and then tomorrow during block, we'll talk about protein synthesis.